Good afternoon, everyone. I hope you are all doing well. And also, I just said good afternoon, good evening, good morning, depending on where you are joining us from around the world, because I know we have a diverse audience from all over the world. Thank you so much for joining us, and welcome to yet another edition of the GSU Malaria Gen monthly series. It's a journal club that we organize on a monthly basis, or try to do so. So far, we've had about 9 or 10 journal clubs. Please do check our Malaria Gen website and YouTube channel for the previous ones. Today's webinar is a vector-focused one, referencing Cynthia's recent preprint titled, as you can see on the screen, Key Gene Modules and Hub Genes Associated with Pyrethroid and Organophosphate Resistance in Anopheles Mosquitoes, a Systems Biology Approach. My name is Pablo Chauke, for those of you that don't know me, and I'm a training coordinator at Malaria Gen, working closely with a team of awesome people to organize these monthly events. Just a few ground rules. The Journal Club is being recorded, as I said earlier, and will be available on our Malaria Gen YouTube channel. So please, after a few hours today, do check out our YouTube channel for the recording so that you can rewatch the Journal Club and share with your colleagues and networks as well. Kindly stay on mute so that we don't disturb our speaker. We will have a Q&A session at the end where you can raise your hand or type your question or comments in the chat, as well as the Q&A box. So while Cynthia is presenting, you can type in the chat if it's a burning question, and she'll answer it at the end of her presentation. Without further ado, I would like to introduce our speaker for today, Cynthia Awuor Odhiambo, all the way in Kisumu in Kenya, who kindly agreed to give this webinar when I invited her a few months ago. And I just want to also brag, Cynthia is a graduate of our Anopheles Genomic Surveillance Data Training course from the 2023 cohort. So yeah, we do know her quite closely, and she's one of the dedicated scientists that we've had the pleasure and experience of interacting with. The title of the talk, as you can see on the screen, is similar to the preprint that's out. I'm just kind of gonna read a bit of the short bio about Cynthia and who she is and the work that she does, and then she can begin her talk. Cynthia Awuor Odhiambo is a researcher and a master's student doing an MSc in molecular biology as well as bioinformatics at the Kenya Medical Research Institute, or KEMRI for short, at the Center for Global Health Entomology Department. Through her work with the Genomics of African Vectors for NMCP Management of Insecticide Resistance project, she is part of a team using whole genome data to support the identification of insecticide resistance markers in malaria vectors for public health decision-making within National Malaria Control Programs, or NMCPs for short, in countries such as Benin and Kenya. In this journal club, Cynthia will discuss findings in a research where she was a first author in the preprint, which uses a systems biology approach to identify insecticide-resistant markers in 2 mosquito species. Cynthia, thank you so much for joining us. You may begin your presentation.
Thank you very much, Pablo, for introducing me today. And thank you everyone for joining the webinar and the journal club today. Honored to be presenting our recent preprint on the work that we've done on identification of different insecticide resistant markers in Kenya and even in Benin. So today I'm going to present to you this manuscript preprint that is currently online, and I'm going to represent the whole team that we've been working with together in helping in identification of these markers. So my topic is just like the preprint topic. So key gene modules and hub genes associated with pyrethroid and organophosphate resistance in Anopheles mosquitoes. This is by using a system biology approach. So I hope I'm going to touch on I'm going to introduce on how the system biology approach was used here and how we did this and the results that we got from this analysis. So just for introduction, I hope everyone here is very familiar with malaria transmission. So we know that the mosquito or Anopheles vectors are the vectors that are transmit malaria from one person to another. So we know malaria transmission starts from one host to another by a bite from a mosquito. So to prevent this, several tools have been used before to help in elimination or to prevent transmission of malaria from one host to another. So some of these tools that have been used, or most of them, include the use of insecticides. So for example, the insecticide treated nets that we use, the ATSB, some of them of these tools have insecticides in them, which help in mortality of the mosquitoes and leading to reduction in the number of mosquitoes, hence reducing the malaria transmission. So in use of this insecticides has led to selection pressure on the mosquitoes, and they've ended up mutating, which caused resistance in the mosquitoes. So this is where now we have the problem of resistance, and there is a lot of issues that has come up with the emergence of resistance across Africa and even internationally. So the problem, I'll start with this paper that has been recently published. You can see the effect, this is from 2017, the effect of genetic resistant mechanisms. So you can see from the map, it has led to a lot of resistance, which lead to inaccuracy affecting the efficacy of interventions that we have. For example, the treatment of malaria by increasing the malaria cases in different regions because of the high number of mosquitoes in different regions. So this has led to inefficacy of our interventions and has led to even affecting the intervention, hence they're not working anymore so good. And even from last year, from the WHO reports, you've seen that there is no much difference in reduction of malaria crisis, and there was an increase in malaria infections from the last year WHO. So the main issue, that many more different problems that have caused this change, but one of them is resistance, not only on the vector side, but also on the plasmodium side. So in this project where we were working towards identifying molecular markers, we had several questions that we were working towards clarifying and even getting answers to. So the first question was, how do mosquitoes develop resistance to insecticides? That was the first question. And the second one was, what methods can we use actually to detect these resistant mosquitoes? And the last one was, which strategies can we use to enhance the effectiveness of these insecticides? So in all these questions, we had an idea of what we wanted to know from them that could help us or even inform us as to what to do to make the insecticide effective, and also improve on our tools that we already have. So to answer this, we developed we designed an experiment or a project that could help in using RNA-seq to solve or to get a solution to all these problems. And in the first question, when we came to think about it, we knew that one thing that we needed to understand how development of resistance was to understand the molecular mechanism. And the second one was to identify the markers, and the third one was to identify the targets. So in all this, we found that we could use system biology to answer all these questions at a go, which was a plus other than doing different researches from different research or different objectives here and there. But we could use this one tool to answer all these three questions at one. So what is system biology? If you're not familiar enough about it, system biology is just, it's a field, a study that focuses on the complex interactions of the biological system. It aims in just understanding how biological systems interact at a molecular level, or how they interact to give rise to a particular function or a behavior of a system. So it's actually how it helps us understand the big picture of what's happening in a mosquito or in a human being or any system that we are working with. So that's why we opted to use it to try and understand resistance, at the same time identify the targets and or even the markers. So we used weighted gene co-expression network analysis, which is a tool, a system biology tool that helps us create gene co-expression networks, and also help us in identifying the most connected genes in those networks. And weighted gene co-expression networks, we used it because it uses RNA-seq data to look or to study the patterns of the genes, how they're expressed in different conditions. And by learning through those patterns, it groups those genes together and help us understand their main role in the system. So why did we not use the standard methods like the differential gene expression network, differential gene expression rather? The thing was, in such standard methods, we mostly analyze the, we analyze the programs. Just a minute. So most of the standard methods, we analyze them in each isolation of each gene, whereby we look at how one specific gene or different genes are just expressed at their own. We don't consider how they work together with others to cause that effect in the system. So that is one thing that was not portrayed in this method. In the previous method, the standard method, which is gene differential expression system. So in using system biology, we are able to study how the genes behave together. Because ideally, mostly we know that proteins don't work together, they don't work alone. They work in together to cause a different function to happen. So that is the idea that the system biology uses to help us know the specific genes that are causing the effect on the condition and a particular condition. So these changes can go across different conditions, and by learning them, they give us important insights, such as the molecular mechanisms that we can learn from. So another aspect that the standard method was not giving us is identification of hub genes. When we use system biology, we are able to identify the highly connected gene in the system, which shows that this is the gene that is have the superpower in the system to help the others, or to, it has a more effect. Without it, maybe the expression of the other genes will also go lower. With it, it goes higher. So that is a very important gene in the system. And in system biology, these hub genes give us more intel. We get that from them, which we are not able to get from differential gene expression. In differential gene expression, we'll just be able to get the highly and differentially expressed, highly or lowly expressed genes, but we'll not know their specific effects on the group or the other groups that are differentially expressed. So how do network work in a simple term? So for example, when you're in a webinar like this, so people have different reactions in such a conference. Some of them will be excited, some will be bored. So using this system biology process, we'll just not assume that the only excited ones are the only ones who actually find this talk interesting, but sometimes maybe the bored ones are also having an effect on the excited ones. So we work it out by grouping these people first. We study them together. We look at, group them according to their profession, where they come from, for example, students, the senior researchers, university staff, etc. So after grouping them, from those groups, we'll be able to identify that one connected person that actually is the foundation of the others, that is causing or have more influence in that network. So when we create a network, we'll be able to find that person. Without a network, we'll be analyzing these people individually, which won't bring the effect that some of them have in the system. So we'll be able to know that person that is actually, sometimes you might find it's the funder who is the most connected. It's connected to different researchers, different universities, etc. So this hub gene, this most connected person, we call it the hub gene in the system. So in simple terms, that is how the network works. So when we come back to the genomics part, where we use the RNA-seq, we have a mosquito. So when this mosquito is exposed to different insecticides or an insecticide, the genes will react differently. Some of them will be highly expressed, some of them will be lowly expressed. And just as I have said, we can't assume that the genes work individually. They work together as a group to cause a particular condition or function to happen in the system. So when we have these genes, what weighted gene co-expression network does is group them by studying their pattern of expression. So when it groups them, we'll get an clustering, an hierarchical clustering of different genes and how with similar patterns. So if they have a similar pattern, they go up together, they go down together. So we'll have those genes grouped together, and after using that, we'll use that to create a weighted gene co-expression network, a network that have the most connected genes. We can be able to identify the most connected genes, which are the hub genes, and be able to know the specific group of genes that have the that are actually causing the condition of resistance or another different thing. So this is what we did. This system biology has been used a lot in cancer and even COVID-19 research to identify markers, and these have been specific and very useful markers and targets for even the COVID-19. So we thought that this will be a very good method to use when identifying markers, not only for resistance or even for effectivity in the ATC. So what we did is we went and collected samples as we collaborate. We are not only doing this in Kenya, but also in Benin. We collected larval samples from the field. Then we reared them to adults and then exposed them to different insecticides. So we exposed them to alpha-cypermethrin, deltamethrin, and primifosmethyl. Then after getting the resistant and even the susceptible samples from the bioassay, we sequenced them at CDC Atlanta, and then we analyzed the results and did validation using real-time PCR just to see if whatever we are seeing from our results it's the same thing that we got from our RNA-seq results. So that is the methodology we used. And I'll start by the first results that I can show here from Helga, one of our co-authors in the manuscript. We had, since we were doing the bioassay and we wanted the resistant mosquitoes, we wanted them at the highest, the most resistant. So for example, in this case of alpha-cypermethrin, we had two sites, Bacilla and Jogu. So in alpha-cypermethrin, for example, we used the most resistant. So in this case, we used the 5X samples as the most resistant. As you can see, they are around 90%, 90% mortality rate. So the 10% that we got from this are the ones that we considered the resistant one. The same we did with deltamethrin, where we used the most resistant. So for example, in 5X, we could not find enough, most of them were susceptible. So we used at 2X for deltamethrin. We did the same for both sides and used the most resistant. For Anopheles arabiensis, we did the same as quoted here by Moke et al. 2024. So we had two sites here in Migori. This one was from Kenya. And we took the most resistant samples at the highest concentration that we could get. So like for CIA, for alpha-cypermethrin, we used 1.5. We took the 1.5 samples. And for Delta, we used the 1.5X samples. And for primifosmethyl, we used the 2X like that, and even for Migori. So it is worth noting that the bioassays, we did them, we exposed the samples for 30 minutes and immediately just took the knocked down ones we left, we preserved them differently. And then we just took the survivors from the 30-minute exposure and preserved them in RNA lighter, which later on the samples were extracted and sequenced. So from the two data sets, one from Benin and the other from Kenya, Anopheles gambia and Anopheles arabiensis, we had both susceptible and resistant groups. And from these two plots, you could clearly see there was a difference between the susceptible and even the resistant, which means even when we are running our weighted gene co-expression network analysis, there will be differences between the expression levels. So we went ahead and did our weighted gene co-expression network analysis. The first thing that we did was the hierarchical clustering. This is what I was trying to explain, like the genes are grouped together according to the expression levels patterns. So when they are grouped together, we get different colors. These colors just means different set of groups, different sets of genes working together. It is unfortunate that in WGCNA, we don't have clear labeling of group of genes together, but we are still using colors to represent that. I hope we'll keep on following. So those are the genes, and down here we have the modules, which are the colors over here. So the difference between dynamic tree cut and the merged dynamic is just that after the dynamic tree cut cuts the tree, it's automated, but sometimes when you look at them clearly, for example, the first three, you can see they really look familiar. The expression looks familiar, sorry, they look similar. So when we do merge dynamic to merge any closely related patterns together. So they'll be merged together to form one big module. So we did that, and we were able to get our specific modules that we did more analysis to understand what are they and what happens to those groups in both Anopheles gambiae and Anopheles arabiensis. So the next step was to identify what happens with these modules, which modules are actually changing across resistant and susceptible networks. So in system biology, we have the Z summary function that helps us calculate or even compare two different networks. For example, in this case, we had a resistant and a susceptible network. So it helps us compare these two networks and be able to identify those modules that are preserved in the resistant network. We were able to identify those, and you can see from the first plot, the Gambi plot, you can see the green yellow was very different, was highly preserved compared to the others. And we had the black, the salmon, yellow, etc. So to view this, we did the same for Anopheles arabiensis, and we had the yellow, the brown for dark magenta, and even yellow-green. At this moment, maybe it might be confusing, but they're still colors. But when you go ahead and look at the specific genes inside those modules, we'll be able to make sense why they are preserved in those networks. So the next thing we did was to visualize the specific modules. So we selected the top 4 preserved modules, for example, green, yellow, black, salmon, and even royal blue. And on the Anopheles arabiensis, we selected yellow, brown, yellow, green, and even dark magenta, just to visualize how preserved they are, how differential they are, how differentially expressed they are in the different data set, in susceptible and even in resistant. So we did that, and you can clearly see there is a huge difference between the susceptible and resistant ones. So in all the preserved modules, we could clearly visualize difference in expression levels. And from this, you can at least see that when you use differential gene expression, some of these genes will be counted as the top 10 or top 20 differentially expressed gene, but not all of them sometimes are reported. But the idea is they work together to cause a specific function. For example, in this case, the resistance. So we went ahead to look at the specific genes that were in this group, mostly looking at the most connected gene in those modules, which was of great importance. So in Anopheles gambiae, when we started with the first module, we had zinc finger protein as the most connected gene in that module, which in this case, as we know, zinc fingers, a lot of those proteins are used in binded to transcription factors. So ideally, the genes that were in this module were the ones that help in gene expression and regulation, since there was a lot of genes that are expressed and some are here because of the effect of insecticide. So the next one, the second preserved module in Anopheles gambiae had E3 ubiquitin protein ligase as the top or most connected gene in that module. And E3 ubiquitin is a known enzyme that helps in breakdown of different proteins. And with the assumption that its role in breakdown of different proteins, assume that it might be one very connected to the breakdown of insecticides while in the system. The next one was angiotensin converting protein, which is another regulatory associated protein. And the last one, or the fourth one, was serine protein kinase, which is mostly associated with immune reactions and even, a lot of immune reaction. It is worth noting that in these samples, we saw a lot of immune proteins. We identified a lot of immune proteins that were different, that were in the samples, and which led us to this idea that maybe even the immune reactions might be part of helping in the mosquito survive insecticide at some point. In Anopheles arabiensis, we had, as you can see, this protein is the top connected, the most connected gene in this module, yet it is not well annotated. This is quite familiar in Anopheles arabiensis. The annotation sometimes is not that good compared to Anopheles gambiae. So we had a lot of unspecific products or not well annotated proteins identified as the most connected genes in this system, giving us the idea that we really need to do more annotation in other species and even look at things deeply in the Anopheles arabiensis. The next one was the protein chiffon, which is also another protein in the regulation, helping in gene expression and regulation. And we got another unspecified protein as from highly preserved module. And the last one that I think most of us are really familiar with, and there was a lot have been done and proved that GST from even differential gene expression, a lot of GSTs have been seen as a target. Yeah. So we saw all these hub genes that were in the preserved modules, meaning we really need to look for them, look for the transcription factors associated with them and try to understand what specific role they have and see if we can use that to make better insecticides. So the next thing was to identify the hub genes. So we were able to identify the hub genes. So after creating the gene co-expression networks, we identified the hub genes in all the modules that we had here, including the ones that were preserved and unpreserved. And interestingly, we had some genes, some specific hub genes that were in both Anopheles gambiae and Anopheles arabiensis. For example, these were the 4 hub genes that were both in Anopheles arabiensis and even Anopheles gambiae. The first one was the serine, which is associated with a lot of reactions, and even cuticular proteins that was also in the both Anopheles gambiae and Anopheles arabiensis and the rest. So we could see that even in the shared herb genes, we could see some immune proteins in the system, giving us a hint to what's happening to the mosquitoes. The same, just not to ignore our standard method, we had to do differential gene expression for our herb genes to see if they're actually differentially expressed. And in Anopheles arabiensis, we had 6 differentially expressed genes, which encoded some that I've already mentioned, like the cuticular proteins. We had protein chiffon and even other unspecified products. And in Anopheles gambiae, we had 6, also the 6, also including the cuticular proteins, salivary gland proteins, serine protein kinase, and even GSTs. So this gives us the validation that even the herb genes that we identified, which in this case are the most connected genes in the system, are also associated with insecticide resistance because they are differentially expressed. Their effect is seen in the system. We did the same for, we did a qPCR just to do the validation further and further. And interestingly, in the qPCR, the expression levels were higher than the ones that we got from the RNA-seq data set. And we were comparing between the susceptible and the resistant, just as we were doing with the RNA-seq, the differential gene expression. So this is quite clear that whatever we were seeing from the RNA-seq data actually reflects on whatever we found on the RNA-seq data on the qPCR. And showing that these are potential markers that can be used for resistance or for insecticide resistance. The next thing that we were looking for in the system was trying to understand the molecular mechanisms. So what we did was to identify the most enriched molecular mechanisms in the system. We used David tool to do this, and then visualize the network in Cytoscape. And in Cytoscape, we were able to see the most enriched pathways and how they are connected in the system. For example, our first or the biggest molecular mechanism that was enriched was the transmembrane. And this was expected because the transmembrane actually carries most of our target sites. Our insecticide targets are in the transmembrane. So there was a lot of happening at molecular mechanisms around that area. And this was connected to innate immunity, the immune proteins, and even was connected to this. So there was some signal from the transmembrane leading to the signal leading to innate immunity reactions happening. Another molecular mechanism that was highly enriched was the signal transduction, which makes sense in the form that there is a lot of signal from the transmembrane region that causing leading to signal transduction on the other side. And another big or enriched molecular mechanism was the metabolic pathways, which in this idea, we know that there is a lot of breakdown of the insecticide that has actually helped in the mosquitoes withstanding the insecticide and even being resistant to it. And the other ones were DNA replication, fatty acid degradation, and even protein degradation. So all these molecular mechanisms revealed to us, or was a representation of what's happening in that resistant mosquito at that particular point, and was showing us the different things that are happening to that mosquito that are making it a little bit resistant and active. So we went and did a summary of what those IR mechanisms are and where they are located in the system. So for example, when the mosquito digest insecticide, they get through the odorant, the cuticle, and the odorants bind to the odorant binding proteins, leading to them being attached or being signaled through the olfactory sensory neuron, and going ahead to our targets, which is going through the transmembranes. So when they reach the transmembrane, different things happen. For example, one thing that happened is there is a lot of signals sent to the CNS by conversion of the glutamate to GSH. So this causes a lot of signal to the CNS, send a lot of the signal to the CNS. And there is a lot of DNA replication and even gene expression and regulation happening, which shows a lot of nucleus activity happening. And this is all related to from the target site up to leading to a lot of replication and even expression of genes towards the effect of the insecticide. Another signal was, you can see here, there's a lot of oxidative phosphorylation happening. This is because of the stress that the cell is undergoing because of the induction of insecticide in the system, and more like the proteasome and even innate immunity, a lot of happening in the system. This gave us a detailed view of what the mechanisms are and everything that is happening. And it's good to know that this was similar in both Anopheles gambiae and Anopheles arabiensis for the two species. And this was a representation of everything that we saw from understanding, using the system biology to understand the mechanisms. So in conclusion, we could say that WGCNA was a very effective tool in identification of potential markers, and even it helped us in understanding insecticide resistance. And some of these proteins we identified here are actually potential markers according to how they're differentially expressed in the system. And it's also good to note that from this, we could see that it's not only a specific reaction that is causing resistance. There are different things that are happening molecularly or cellularly. The neural signaling, etc., the immune modulation, even gene expression, all this happening at the same time. And we need to note that and use them in producing new insecticides. And into recommendation, we still need to go to the next step and do functional validation for these hub genes, especially the ones that were in both Anopheles gambiae and arabiensis, and do functional validation for them. And also, we would like to recommend that consideration of these mechanisms in creating new insecticides, which will be more effective in the future. And lastly, we'd like to also recommend that this can be used in assessing and even combating insecticide resistance by the NCPs, these markers to be used in identification of resistant mosquitoes. And I would like to acknowledge or thank the entire genomics team, including our section head, Dr. Eric Ochoma, and even our collaborators from CDC, from Benin, from Arizona, and the NMCP, and everyone at large, and even the Gates Foundation for funding this project. Thank you very much. Any questions?
Thank you so much, Cynthia. This was brilliant, really informative, well-explained presentation. Thank you so much. It's now time for Q&A as well as discussion. So please kindly raise your hand or type your question in the chat or in the Q&A box, and we'll be able to ask Cynthia to engage us. We have about 20 minutes to go, so we have a bit of time for questions. While waiting, I'll start, and my question might be a bit generic, but it's something that I constantly think about. It's achieving zero malaria in our lifetime, hopefully in this generation. So maybe some of us might not be alive then, but the point is, I think we are working towards trying to end malaria. And I think you spoke a lot about insecticide resistance. And I think that obviously is taking us further back in achieving zero malaria. From your experience in your study, do you think we could achieve zero malaria in this generation, even with insecticide resistance?
Okay. Yeah, thank you very much, Pablo. Yes, I think it's possible. Maybe it's being overambitious sometimes, but yes, I think it's possible. With the use of the current, in Africa, we are currently doing a lot of genomics work, which I think will bring a lot of effect in early detection of this resistance and even other issues, the genomic part mutation and a lot of things. So when that is detected early enough, I think, yes, we will be able to be at a point where we say zero malaria. We don't have any malaria across Africa. Yes, so the genomics work that we are doing across Africa will bring a lot of effect on leading to that goal. Yeah, thanks.
Thank you so much for your answer, Cynthia. And I like the optimism. Deriba in the comments says, great work. Ilia Mdam says, great presentation. Congratulations, Cynthia, for your work. Your work impacting on control of malaria in Africa. That's from Ilia. As I say, we have a bit of time for questions and comments. So please kindly type in the chat or raise your hand if you would like to speak, and we can let you use your mic. There's a question from someone saying, please explain how the transmembrane pathway is contributing to resistance. The question is from Hev. Did you get that, Cynthia?
Yes, I can see it from this side. Okay, so the transmembrane is where most of our insecticide targets. So there is a lot of target resistance coming in, like the knockdown, the KDR. It targets where the resistance happens. So it's the target. So when the insecticides get into the specific places, there is a lot of mutation. The mosquito try to mutate that particular target so that the effect is not as the one that you think it's going to happen to it in layman language. So that is how it influences or even helps in contribution to resistance by causing there is a lot of target resistance happening, showing that.
Hey, thank you so much, Cynthia. There's about 2 hands raised. I see Hev Raul Tazokong. Please go ahead and ask your question, Hev.
Thank you very much, Babalo. Thank you very much, Cynthia, for this nice piece of work. So I would like to know from the various comparisons that you did, because you showed a volcano, and I wanted to know, how do you proceed to compare your different samples? Because you mentioned that in the phenotypic resistance, you consider the knockdown resistance at 30 minutes. So I wanted to know if the susceptible, as you use here, was the one that was knocked down, and then the resistant was the one that did not knock down during the phenotypic, and then you compare, then you did a pairwise comparison, or you compare all of them versus a Kisumu. That is my first question. And then the second one is, I wanted to know what is really the main difference between the system biology approach versus the pathway enrichment analysis after conducting the differential expression gene analysis. And the last question is about the main insecticide resistance mechanism from Anopheles gambia species. We know that we have mainly target site, but secondly, we have metabolic resistance. But from the main gene that you found involved in the hub, they were mainly from other different biological pathway, apart from cuticula, which is already validated for insecticide resistance. What do you think about this? Was this result was expected, or it was something that you did not expect to see? Thank you.
Okay, thank you very much for the questions. So yeah, for the first questions about the comparison, when we created the volcano plots, we compared the resistant and the susceptible, which is the susceptible group. In this case, we were using Kisumu strain, which is a non-susceptible strain used as control in different assays. So we used Kisumu strain, and the same for Arabiensis, we used susceptible strain to compare and get the different volcano plots. So in the volcano plots, you can realize that those were, the red ones were just the specific hub genes, and we tried to ignore the different, the highly expressed ones. So we were just focusing on the hub genes in this case to see if they are differentially expressed. On the next question, you are talking about the pathway analysis that you do after differential expression, if it's different from the one that we did here. Just as I explained before, the difference between these two pathways is one is just focusing on the differentially expressed specific genes that are top in the system, but the other one is representing everything that is happening, including generation or DNA replication, and even the factors that are causing production of different genes along the way, the differential expression. So I think they're not that different, but the difference is just one is a group of genes, the other one is just the top genes. I don't know if you get that. And for the last question, you asked about in the hub genes, we got some that were not the metabolic genes or the metabolic ones, or even that we got the target site and etc. So these different ones, that is now where the question comes in. Since we had some new genes that we have not seen before, and according to this system, we have seen that they have a function in the RNA, in the insecticide resistance pathway. So the question is, what we want to do next is the functional validation to actually see if the function is seen from this. So I can't say that they are not there or they have not been discovered before, but maybe their function haven't been seen before because we've been looking at them individually, the different expression of genes. So I could say when we did this, the use of differential gene expression was a way for us to try and validate if we are on the right track, which we are, because we could see the cuticular proteins, etc. But also finding the other new markers, that was also a good thing in this case, since, yeah, I think they had not been seen before, but they have a critical role in insecticide resistance. Yes, that's what I can say. Thank you.
Yes, sure. Thank you so much, Cynthia. We have a couple of questions coming in. I see a hand from Dennis. I don't know if, Dennis, you want to go ahead and ask a question, or if I can read from the chat. Dennis?
Yes, can you hear me?
Yes, we can hear you.
Okay, my question is, does the presence of the genes shows that they are resistant? And the next one is, what is the difference between system biology and the normal bioinformatics work we do? Like the one the normal bioinformatics tools we use.
Thank you for the question. So I try to show a difference between the two here. I just try to. So the normal way we use is usually doing the differential gene expression, which just focuses on the top differentially expressed. So we use them as the markers. But when we come to think of it, they are the genes that actually are causing those effects to happen, making one to go higher than the other. Expression of one gene is affected by the expression of different other genes. So in system biology, that is what we are trying to see compared to what we usually do. We are trying to see how these different grouping of these different genes, how they work together to cause resistance. And you can see that after identifying these modules, there was actually a particular pattern they had in resistant and even in susceptible assays. I think this was the slide here. So I said like the differential gene expression, mostly it's just checking and taking the top. You're just working with the layer, but you're not focusing on maybe the background. You might find, like you can see from the results that we have found, there are some cuticular proteins that are actually causing other cuticular proteins or other metabolic proteins to have that effect of going extremely higher in the network. This is what the normal analysis or the differential gene expression is not showing us. And it's something that is really informative and insightful in understanding different biological system or even condition. Yeah. And you asked about having the genes. Does having the genes mean that they're resistant? Did you ask it like that, or what was your specific question?
Yes, when maybe we see the resistant genes, so do we like conclude that they are resistant?
Yes, like no, in this particular point, I can't assume that, but we have to do functional validation to actually see what's happening and to see if we see this particular gene at this specific level, is the mosquito resistant or not? We have to validate that for us to conclude that these genes are actually showing resistant mosquito. This method showed us a way to go into and look for this specific gene, this specific cuticular protein, this specific metabolic proteins. We have to look deeply into them and do functional validation for them. Yeah.
Thank you. Thank you.
Thank you.
Thank you so much. I'm looking at the time, and sadly, we've gone over time, but I can squeeze in just one last question. And for those that have their questions not answered, please do get in touch with Cynthia via email or on Twitter. The question that I'm going to ask you is from Messi Tabamifo, who says, great presentation, thank you. Please, after the bioassay, how was the mosquito preserved before RNA-seq? That's from Messi. Cynthia?
Okay, thank you, Mercy. So the mosquitoes were preserved in RNA later. So immediately after we identified or we got the ones that survived, the survivors, we removed them from the particular bottle and then aspirate them to a new paper cup. And that paper cup, we knocked them down in the freezer and immediately put them in separate tubes and added RNA later inside. And later, those are the ones that we've used for sequencing. We'll sequence them later on. Yeah.
Okay, thank you so much, Cynthia, for that great presentation. I just shared Cynthia's email in the chat for everyone. If you want to email her and ask your questions directly, please do. I asked her for consent in the beginning. She's happy to receive your emails and for you to follow her on Twitter and engage further on this preprint. Cynthia, this was a brilliant presentation, and everyone in the comments keeps on repeating that. Great presentation, Cynthia. So thank you so much for your time and graciously agreeing to give this presentation. We really learned a lot. To all of you who attended today, thank you so much for attending our June Journal Club, GSU Journal Club. We are going to run another one in July. Just be on the lookout for adverts on that, on our social media, our newsletter, and our website. And as I said, this talk was being recorded and is going to be available on the Malaria Gen YouTube channel later today. Thank you so much, everyone. Enjoy your day, enjoy your evening, depending on where you are in the world. Cynthia, we are really grateful. Thank you so much, everyone.
Thank you very much, Pabalo, for having me. Thank you.